Hello viewers, welcome all of you in Alka's Botany channel. Today we are going to learn Penicillium, an important fungi for the human life. I am Dr. Alka Inamdr in Alka's Botany channel. Please like, share and subscribe the channel. In the classification of Penicillium, it comes under domain Eukaryota, kingdom fungi. Eumycophyta is a division of Penicillium, class Ascomycete, subclass Euskomycetes, order Aspergillus, family Aspergillaceae, and genus Penicillium. In the habitat and occurrence, the genus Penicillium is a cosmopolitan, found at various places such as soil, air, and on decaying food material, etc. Penicillium is a genus of saprophytic fungi, that is, it feeding on dead and decaying matter. They are commonly known as blue or green mould. They play an important role as a decomposer in the ecosystem. Penicillium means painter's brush. They are named so due to the presence of chains of Conidia produced asexually appearing as a brush terminally on the mycelia. Penicillium expansum is a parasite causes rotting and spoilage of fruits such as apples, pears, grapes, etc. when they are in the storage position. Penicillium notatum and Penicillium chrysogeneum are used for extraction of antibiotic penicillin, which is an important research for the human life. In the structure of mycelium, it shows a vegetative structure which is multicellular mycelium. The mycelium is made up of highly branched, multinucleated and septate, long thread-like filamentous structure known as hyphae. The cell wall is made up of glucose, polysaccharide and chitin. The cytoplasmic continuity is maintained through central pore present in the septa. Some of the mycelia grow deeper into the substratum to derive food material, and the food material is stored in the form of oil globules. In the reproduction, Penicillium reproduces by vegetative method, asexual method, and sexual reproduction. In the vegetative reproduction, first one important method is a fragmentation. The hyphae breaks up into a short segments that each segment is called a fragment, and that fragment grows by repeated division into the full-fledged mycelium. Second one important method of vegetative reproduction is sclerotia formation. In some species, the mycelium forms compact resting bodies. These are called sclerotia. These sclerotia enables the species to survive periods of stress or to hibernate. On the onset of conditions favorable for growth, each sclerotium germinates to form a new mycelium. In the asexual reproduction, which takes place in the spatial structures which are non-motile asexual spores called Conidia, which are produced on conidiophore. Conidiophore may be unbranched or branched and differentiated into matulae. Number of flask-shaped phyllite cells, or it is also called sterigmata cells, develop at the end of each matulae, which forms conidia. Phyllite cells or sterigmata cells divide mitotically, forming a conidium. Each phyllite cells again divide, pushing the first conidium towards outside, and the second conidium is formed. This process repeats, resulting in a chain of conidia found in a basipetal succession. That means younger one at the base of that chain that is near to the sterigmata, whereas older one at the outside of that sterigmata. The conidia may be blue, green or yellow in color and oval or elliptical in shape. Conidia get detached from the parent after maturation and dispersed by the wind. They get attached to a suitable substratum and they get germinate. Structure and germination of conidia, in which the conidia are tiny spore-like structure, globose to ovoid in form. The spore wall is differentiated into two layers, outer exine and inner intine. The exine is comparatively thick, smooth or spiny, and the intine is very thin in structure. On falling on a suitable substratum and under suitable condition means a moisture, suitable temperature and food material present on that substratum, and the conidium absorbs moisture and swells. Germination is by the germ tube formation. The nucleus divides mitotically multiple times. All the nuclei enter the germ tube and followed by the elongation and formation of septa to give rise to a mature and branched mycelium, which is easily seen in this diagram. Under sexual reproduction, it is by the formation of ascospores. Some species of Penicillium are homothallic and some are heterothallic. Homothallic means male and female sex organs develop on the same thallus or in the same hyphae, then we call them as a homothallic. And heterothallic means one hyphae or one filament of Penicillium develops male gamete, whereas another hyphae develops female sex organ. Then we call them as a heterothallic. The male and female sex organs are Antheridium and Ascogonium respectively. Ascogonium, which is developed first sex organ than the Antheridium. The mature Ascogonium is a long, erect, multinucleate, unseptate tubular structure which is seen in the diagram. It arises as a lateral outgrowth from any cell of the vegetative mycelium. When young Ascogonium is uninucleate, it is elongated. The single nucleus within it divides and redivides to give rise to a definite number of daughter nuclei, which is either 32 or 64. Antheridium. Meanwhile, a slender uninucleate hyphal branch originates either from the adjacent cell of the same hyphae, which develops ascogonium, which gives rise to a ascogonium, or from a separate neighboring hypha. It is the antheridial branch which is seen in the diagram. It grows up coiling loosely around the ascogonium, making several turns around it. The distal end of the male branch becomes slightly inflated and is eventually cut off as an antheridium by a septum. The antheridium is a short, terminal, club-shaped uninucleate structure. After that, important process that is a plasmogamy takes place in the antheridium and ascogonium. It is the union of two protoplasts which brings the compatible nuclei close together in the same cell. The tip of the antheridium comes in contact with the ascogonium. At the point of contact, the double wall dissolves and through the common pore, the two protoplasts come in contact. The male nucleus migrates into the ascogonium. It divides a number of times. The male nuclei then come to lie by the side of the female nuclei, each to each. Each pair of nuclei consisting of one male and the other female is called dikaryon. Di means two, karyon means nuclei. Two nuclei simultaneously in the pairing position. Post-plasmogamy or autogamic changes. After plasmogamy, what happens? There is a first important phase is the septation of ascogonium. Plasmogamy or autogamy is followed by septation of the ascogonium. Each segment has a pair of nuclei. Meanwhile, entangled sterile hyphae grow up around the sexual apparatus and form an investment of loosely interwoven hyphae and sexual apparatus. Second one stage is the development of ascogenous hyphae. One or more lateral outgrowths arises from some of the binucleate segments situated in the middle of the septate ascogonium. Each outgrowth is called as ascogenous initial. It is a binucleate. Each ascogenous initial develops into a branched ascogenous hypha composed of binucleate cells. Third one important phase is the formation of asexually. Asexually in many species are developed directly from the binucleate cells towards the tip of the branches of the ascogenous hyphae. Almost all the cells of the ascogenous hyphae develop into asexually. Asexually is the plural form, whereas singular it is called as ascus. And due to that formation, cleistothecium, which is a fruiting body, develops. The peridium or sheath is a thicker and consists of loosely interwoven hypha. This entire structure is a spherical and has no opening. Such a closed fruit or ascocar is called the cleistothecium. Cleisto means closed and thecium means fruiting body. The asci are born in chains within the ascocar. The terminal dikaryotic cell swells up forming ascus mother cell. Then karyogamy or fusion of two nuclei occurs in the cell forming the diploid cell. While in the previous form there is a pairing happens and these two nuclei, one is of male nucleus and another is of a female nucleus, they fuse to each other to form a karyogamy. The diploid zygote undergoes first meiosis and then mitosis to form 8 nuclei. Each accumulates some cytoplasm, resulting in the formation of 8 ascospores. The ascospores are released by the dissolution of ascus wall and get attached to a suitable substratum. In each ascus, there are 8 ascospores are developed by first meiosis and then mitosis division. The ascospores develop by forming a germ tube and mature into the branched mycelium. This is the graphical life cycle of Penicillium, in which it shows Penicillium mycelium asexually reproduces by means of conidiophore. On that conidiophore, Conidia develops and this Conidia germinates to form again a Penicillium mycelium. This Penicillium develops ascogonium and Antheridium as a sex organs. After that, pairing happens with paired nuclei. After that, this ascogenous hyphae develops into a karyogamy, and after that karyogamy, young ascus develops. These asci, when interwoven by the sterile hyphae, Then there is a closed fruiting body called a cleistothecium develops. In each ascus, there are 8 ascospores are developed. Each one ascospore by germination develops into a penicillium mycelium. This is a graphical life cycle of penicillium. Then we come up to the last point, that is the economic importance of penicillium. Many species are employed for commercial production of organic acids such as citric acid, fumaric acid, oxalic acid, etc. Penicillium roqueforti and Penicillium camemberti are employed for the production of cheese, whereas Penicillium notatum and Penicillium chrysogeneum are employed for production of antibiotics, that is a penicillin. Penicillium patulum is employed for the production of gentile alcohol. Some species of Penicillium are employed for synthesis of pigments like citrinin in the yellow, imodic acid that is orange color, spinulosin that is a blue purple pigments. Some species are pathogenic, causing spoilage of food material. Example, Penicillium digitum, Penicillium italium damages citrus fruits. Many species deteriorate paper, books, etc. also. Many species causes diseases to animals. Thank you. Thank you very much.